In this video, I will show you how to set up a respirometer. I will also explain to you how that respirometer works in order that you will be able to analyze the data from the respirometer. So let's start with setting up your respirometer. Begin by measuring 50 milliliters of water with a measuring cylinder and pouring into that 25 germinating peas. Make a note of the increase in volume and therefore the final volume of the water. Once you know what your final volume is, you want to separate out the peas from the water, and you can use filter paper to do this if you want. Place your germinating peas to the side and we'll use them later. Now you're going to repeat the process using 25 dried peas instead of germinating peas. So fill up your measuring cylinder with 50 milliliters of water, only this time put in 25 dried peas. You'll notice that the volume doesn't increase as much with the dried peas as it did with the germinating peas, but we want to have the same volume ultimately. So we're going to make it up to the final volume that you recorded with your germinating peas, only we're going to use glass beads to do that. Once you've reached that final volume, remove the dried peas and beads and put them to the side. We'll use them later too. Finally, we're going to repeat this process with glass beads only. So fill up your measuring cylinder with 50 milliliters and then you're going to make it up to that final volume using only the glass beads. Once you've done this, you can put the glass beads to the side as well. At this point, you should have an equal volume of all three. 1 of germinating peas, 2 of dried peas and glass beads, and 3 glass beads only. The first 2 will be a part of the experiment, but the third one, the glass beads only, will serve as a control, which we will use in our data analysis later. Now, before you set up the respirometer, please be aware of the aspects of safety and follow any guidance your teacher provides you with. But at minimum, you should be wearing safety goggles and avoid direct skin contact with the potassium hydroxide that you are about to use. If that contact does occur, please flush the area under running water for 10 minutes. Now, in order to set up the respirometer, you'll need to have a test tube, and at the bottom of that, place a small wad of absorbent cotton. Using a plastic pipette, take 15% potassium hydroxide and place a few drops onto the cotton to saturate it. Be careful not to get any potassium hydroxide onto the sides of the respirometer. Once you've done that, you should place another small wad of dry, non-absorbent cotton onto the top of the cotton ball that you just soaked with potassium hydroxide, and this should protect your living organism, in this case your peas, from the potassium hydroxide. Next, you can put one of your 3 samples into the test tube. In this case, I'm using the dried peas and glass beads. Once you've done that, place the bung into the top of the test tube, and through the bung, you should have already arranged the glass pipette that you're going to use for measurement. Repeat this with all 3 of your samples so that you have 3 respirometers, one with glass beads only, one with dried peas and glass beads, and one with germinating peas. Since you're going to place the respirometers underwater, you're going to need an airtight seal, so I suggest wrapping around the top of the test tube and the bung using parafilm. Now you can place your respirometers into your tray of warm water. However, don't submerge the tip of the pipette yet. You want to give it some time to equilibrate, because if the water is warm, then the gas inside the respirometer will expand and the bubbles will come out, and you want to allow that a period of time to happen before you start your experiment. I would suggest that you leave the test tubes in the warm water for about 10 minutes to allow for sufficient time for equilibration to take place. After about 10 minutes, you can completely submerge the tip of the pipettes. You should see some water enter the tip of the pipette, but it should stop fairly quickly in terms of how far up the pipette it moves. If it doesn't stop, you're going to need to check your respirometer for leaks. Other things to consider are that sometimes the respirometers float, so you might want to attach them to a small weight, as I've done here with the black tape, to make sure that they actually sink. Finally, you need to make sure you can actually read the pipettes. The air bubble should extend from the chamber of the test tube all the way up through the tube of the pipette. You might want to rotate the pipette so that you can actually read the numbers. Finally, you can start the experiment, and what you're going to do is measure the distance travelled by the air bubble in a given period of time. If you look closely in this video, you can see the air bubble moving up the pipette. So how does the respirometer work? During respiration, oxygen is used and carbon dioxide is produced. This results in a change in volume in the gases within the respirometer. But what we really want is to narrow it down to just one of those gases. For example, in this case, the uptake of oxygen. Therefore, we need something to remove the carbon dioxide that's produced. And that's what we use the potassium hydroxide for. So the cotton ball soaked in potassium hydroxide is there so that potassium hydroxide absorbs the carbon dioxide produced from respiration. This means, therefore, that the change in gas volume within the respirometer is then only as a result of the oxygen taken up. Now, because the respirometer is submerged underwater, as the oxygen is taken up for use in respiration, the volume of gas within the respirometer decreases, and therefore the water moves into the pipette of the respirometer. In order to measure the rate of respiration, we can therefore measure the distance or the volume moved by the water into the pipette in a given time. And we need that given time if we're actually going to calculate rate, because rate is the distance divided by the time, or the volume divided by the time in this case. So the rate can be measured in milliliters per minute if you're measuring it using a volume, or centimeters per minute if you're measuring it using a distance. Now, if what you're working on is analysis of data, be that your own data or data you've been given in a question, then see my next video in this series for help with that.